Yeah, recording.
That's wonderful. Thank you. I'm going to wait one more minute as people are still trickling in. But for the people who are already on, the materials are all linked via this tinyurl.com. So if you type tinyurl.com.cmmmcws for workshop, you should be able to get all of today's materials, all the slides and everything that are going to be covered. Hopefully you had a chance to download Cytoscape, because if you haven't downloaded Cytoscape, it will be less useful. You can still watch and learn, but it won't be as useful. So I will start in about a minute from now, and because there are still a couple of people that are trickling in. All righty, we'll get going. First of all, good morning, everyone, at least from San Diego. Good morning, maybe afternoon or evening where you are. Welcome to the third Microbial Metabolite workshop. I'm Professor Dorstein. Helena Russo will be the other presenter, and then Shipe, who's online, and Felipe. If you have any questions, you can ask them via the chat. They will be helping to answer them. Before I go on into the workshop, what the purpose of this workshop is, I want to make sure that we establish the rules. Wait, I got the wrong one here. This is, yeah, I did have the wrong one here. Let me grab the correct one. All right, it's still the same things applies. It's still the same tiny URL CMMC WS. But before I go on, sort of the guidelines, I want to cover the guidelines for today's workshop. They're very simple. Ask questions in the chat, unmute, and ask. Interruptions are encouraged, so you can just unmute yourself, ask questions, or put them in the chat and ask the questions. And if anybody knows the answer to the questions, feel free to respond. We're all here to learn, and the hope in the long run is that we can really improve our ability to understand microbial metabolites. So, and these workshops are an infrastructure that is being built, is really organized by the Collaborative Microbial Metabolite Center. But what is the Collaborative Microbial Metabolite Center? It was a call by the NIH. And the reason why NIH put this call out is because they realized that the microbiome is very important to human health, but it's also important for other ecosystems such as the ocean environment or soils. But what they realized was that you cannot, that it's really difficult to mechanistically understand the microbiome. And one way that you can understand the mechanistic role of the microbiome, and in other words, what are the functions that the microbiome really carries out? That part of the answer lies in the microbial metabolites that they produce. Now, the challenge is that they're not really great resources where you have microbial metabolite information, the organisms that produce them, and their activities that are associated with it. And so they wanted to build a knowledge base and a management center. And then around this management center, there were R01s that are going to be awarded. Many of you that are part of this workshop are contributing to this via the R01s. That all the data from those R01s goes into a centralized place that then can be reused. And so the tool that you're going to learn about today is the first sort of tool to aggregate microbial metabolite information, as well as a way to use it. And so the way it essentially operates, and what you're going to learn about, is that we have a microbial metabolite data portal upload system. The last workshop shows some of the enrichments, but today we're also going to provide an example of an enrichment workflow. We're highlighting how the kind of interface that we now have for upload. And of course, this is one of the things that our laboratory has been doing for a long time, is to enable data reuse and analysis. And we do this in the context of microbial metabolites. There are 6 institutes at NIH that are contributing, so R1 grantees, and many of you that are, as said, that are part of this workshop are beginning to contribute to the microbial metabolite information. But we're also expanding this to the worldwide community. It's a large challenge to capture all this information as a worldwide community. So we're going to really expand it, because that will make sure that we can improve our ability to do mechanistic studies with the microbiome much more effectively. There's now a website. This website shown right here. And there are really 2 key parts for the end user that become important over time. First of all, there are links to the tools that have been created. And then secondly, and one of those tools is an example that we recently submitted a publication for and that we had last workshop on, is Microbial, which basically allows you to search 60,000 LC-MS data sets of cultured microbes. So 60,000 that was curated with the community, so that when you search, let's say, a mass spec feature, you can start to see which organisms are capable of producing this. So you can search unknown and known organisms, known molecules in this fashion. And then we have the workshops, past workshop, the videos of the past workshops, in case you've missed anything. The first one was on molecular networking, to see how you can use that to find microbial metabolites. And the second one was on Microbial, the tool that I just highlighted. The website is very much under construction. So if you have suggestions, we love feedback, all those kind of things. So today, however, we're going to cover 2 of the workshops, or two of the new tools that we have. The first one is the network enrichment workflow, and Helena will go through that step by step with you. And then at the end, I'm going to cover the microbial metabolite knowledge upload portal. And it's not going to be a hands-on portion, but it's going to be actually a call to the community. So because the faster we can populate this information, the easier it becomes to understand the microbiome at the mechanistic level. And this is important because the majority of microbial metabolites are not really documented in sort of traditional metabolic pathways. So if you look at the KEG pathway, a lot of these metabolites that microbes make are just not present in those kind of metabolic pathways. And so this knowledge portal aims to capture that type of information, and then the network enrichment workflow actually uses that information so that you can start to look at metabolomics data and inform about microbial metabolites. Okay, what is the first generation network enrichment workflow? Think of it this way. Let's say you have a fecal sample. This could be a soil sample, an ocean sample, but a fecal sample or a blood sample. And you want to answer these following questions. What metabolites are in the sample? That's just a general question. That's just your standard untargeted metabolomics question. But now go to question 2. Are they microbial? Are they due to microbial metabolism of host, food, or drug-derived metabolites? If they are microbial, what microbes can make these molecules or carry out this transformation? Now, the first one, now there are lots of tools that can answer that question. But the second question and third question are much harder. And so we're going to do the type of data that we're going to analyze here are metabolomics data based on liquid chromatography and MSMS of all the ions that are observed. So you fragment all the ions. And Elena will go a little bit deeper into what that data structure looks like. But then we can apply something that we tend to apply to it is molecular networking. And basically what you get with molecular networking are you can get your relative intensities. You can get annotations from it if you match against spectral libraries. And at this point, the average annotation rate is 14%. And then because of molecular networking, we can get relationships of structural similarity. So you may have a tryptophan here and then an acetyltryptophan next to it. And so you can begin to propagate known and unknown molecules. That's one of the key strengths. But a typical thing that you get is a list like this. And so now if we ask the question, is this a microbial food or drug derived molecule? If it's microbial, what microbes can make this molecule? That list still doesn't help you answer that question. I can even give you the structure itself. It still doesn't help you answer these type of questions. The way to figure it out now is that you have to Google maybe the structure, but if you, let's say, take penicillin, for example, there's maybe 400 synonyms for penicillin alone. So it's going to be so hard to navigate that space. And this is one of the purposes of the knowledge base that we're building so that it becomes easier to interpret what might be a microbial molecule and what might not too. So we have then within our GMPS ecosystem, every analysis that's done gets a task ID. It's essentially a blockchain. And then we can combine this molecular network analysis, and that's what you will be learning today, with the knowledge base that the community has curated, so that you can import it into things like Cytoscape or other third-party tools and quickly recognize what might be a microbial metabolite. So the challenge is that still that most microbial metabolites remain uncharacterized. Right now, the knowledge base itself has grown to about 5,000 annotations of about 600 compounds. And my hope is that in the next year, this will reach a few thousand compounds already. And then in the next, let's say, 5 years, that we're going to be reaching tens of thousands of compounds. And that, I think, to a certain degree, that's pretty realistic. We're just getting off the ground. You just have to think about it. When a baby is born, they don't walk right away. We're just at the stage in the development of this infrastructure where the babies begin to crawl. And so hopefully over the next 2 years, they will reach teenager stage. They go completely crazy. And then over the next 5 to 10 years that it will mature out and that it will become even more useful. And this is one of the reasons at the end of this workshop, we'll have a call to the community and we will highlight how you can contribute. We hope to create a community paper of people who have contributed to it, who will have tested it with their data as examples that we can include into the paper. So the first data set that we will use comes from the American Gut. This is an open platform for microbiome research. Basically, you can pay $100 and your microbiome gets sequenced, and it's a part of an academic program that my colleague Rob Knight runs. Some of these samples we've subjected to a metabolomics analysis. We're going to be analyzing the fecal samples of 2 people here in relationship to microbial metabolites that are there. All righty, I think... Are there any questions from the audience at this point? If not, then Helena will begin the tutorial. So this is one part where you can follow hands-on, so you can sort of understand and navigate the workflow. And feel free to ask any questions at any time.
Okay, so good morning or hello, everyone. So I'm going to be carrying you through the hands-on part of this workshop. And our main goals now will be to launch a molecular networking enrichment job in GNPS2, to inspect these molecular networks in Cytoscape, and map microbial information from CMMC in the molecular networks in Cytoscape. But well, before starting right away, I would like to give a step back, because I'm sure that there might be people in the audience that are hearing for the term of molecular networking for the first time. So I'm going to give a very brief explanation on what does that mean. So let's say that we have a complex sample which contains a mixture of molecules. And in this example here, we have tryptophan derivatives and biotin and distillobiotin. When we analyze this sample in liquid chromatography mass spectrometry, we are going to be generating mass spectrum for each one of these compounds. So each of these mass spectra here, they are very important. They carry very important information about the structure of each of these compounds. So after this spectrum here, they're going to have important information of parts or fragments of these compounds. And it is very well known right now that compounds that are structurally very similar are going to generate very similar fragmentation patterns in LC-MS-MS. So in this case here, we can see that the MS-MS spectra that were generated for the tryptophan derivatives are very similar between each other, but they're very different between compared to biotin and this thiobiotin. And what we can do then is compare how similar these spectra are between each other, and we can do that using the GNPS ecosystem. And we can build what we call molecular networks. So these networks, they are formed by nodes that represent each of these MS-MS spectrum, and by edges that connect this node that show how similar this spectrum are. And here we are showing as cosine similarity, in this case, as closer as it gets to 1, as the more similar that the two spectrum are. And as expected, we can see here in this example that the compounds that are tryptophan derivatives, they are all grouped into one network here in red. And that biotin and this thiobiotin are separated in another network here in green. So as important as comparing spectra that are generated from your samples, comparing your spectra with reference libraries are also very important, because then it can help us interpret our data and really say, okay, what are the compounds that might be present in my sample? And this can also be done by the GNPS ecosystem, as it also has dozens of libraries, of reference libraries populated in there. In the very first...
Does anyone have questions at this point? Because it's really important to understand molecular networking, and if you missed the first workshop, this may need clarifications. If not, great.
So in the very first workshop that we gave as part of this series, we describe in much more details on how these molecular networks are built, how the cosine similarity is calculated, and how you can really launch a molecular networking job and inspect the results. And if you didn't have the chance to attend, we have a hyperlink here. It is also listed. It is also posted on the CMMC website. But this was the molecular networking job that we submitted as this data set that we explored together in the first workshop. As Peter mentioned, it's samples from the American Gut Project, 2 samples from the American Gut Project, and also 3 samples that are relative to microbial monocultures, in which in the media there were bile acids that were added in addition to amino acids. We're going to get more into details later on. But what I would like to highlight here, and Peter already briefly mentioned, is that every job that we submit in GNPS, we are going to have a link here in the browser. And the last part here of this link is what we call a task ID. So this is the task ID for the first workshop that we submitted together. And this is very important to carry on downstream analysis, and you will see that we will be using this task ID soon to submit our enrichment workflow. So let's start on the hands-on part of the workshop. I'd like you to go to the gnps2.org website and click on login on the top right part of the website. I'm going to wait a little bit. And please post on the chat if you're following. You also feel free to unmute yourself if you have any questions.
Is there anybody who is not able to get to this website and log in that wants to follow?
Okay.
Good.
So after you log in, after you click on login, you're going to see a page like this in which you can put your username and password. That is the username and password that I sent you via email last week.
Is there anybody that is on that wants to follow that did not get their username and password? If so, just put it in, put your name and email in the chat, and Shipei will be able to provide it. Great.
Great. Okay, so hopefully many of you already logged in. So after you log in, you can click here on the top left on workflows in the gnps2.org page. And then you're going to see the list of all the workflows that are available in GNPS2. And then you can type in here on the top right, the word enrichment. And after that, you should see just one line after this filter. Okay, and then you can click on launch workflow, and you're going to be redirected to a page like this. So here we are going to be filling and selecting some of the parameters that we are going to be using for the network enrichment workflow. So the very first thing that you should input is the job title. So you can put anything that is that makes sense for you. It's just for your future reference. So for instance, you can put CMMC, workshop, network enrichment. And then we are going to be selecting that we are going to be enriching a workflow, networking, a molecular network that was created in GNPS 1. Are there any questions? Everyone is following? Good.
Yeah. There is a question.
Yeah, what is the difference between GNPS 1 and 2? I was sure that someone was going to ask that. I know that some of you might be a bit confusing. Yeah, I already had a GNPS 1 account. Why are you creating another account for me? So right now we are developing GNPS 2.0, in which we are remodeling how GNPS works, and the workflows that are present in GNPS 1, that is gnps.ucse.edu, are going to be transferred to GNPS 2. So the idea is that it's going to be faster, more efficient. It's basically just a very, very big upgrade that is being made. So, yeah. But right now it's very, it's still in the baby steps as well, as Peter mentioned. So right now we have the two websites that you can use.
Yeah, there will be a transition. In a few years, we'll phase out GNPS 1.0. Currently GNPS 2.0 is being developed. The idea is that we improve the tracking. I mean, the foundation of GNPS 1, it has code from back into 2007. So it has some really, it needs an upgrade basically to be compatible with future faster searches, better tracking of all the data that's in there. So it will be a transition, but for now we're going to use a task ID from GNPS 1.0.
Yeah, so basically it is also possible to launch, for instance, molecular networking jobs in GNPS 2.0.
Yep.
So now you have your login credentials, you can explore that further.
Yeah, and all the tools, all the new tools, including the database that we're using here, are all being built in GMPS 2.0. So the most up to date mass searches, the micromass searches, all of those are in GMPS 2.0 because we just have a better tracking system using universal spectrum identifier. Should you be using GMPS 2.0 going forward? You're welcome to use one or the other. There are some tools that are not yet migrated over to GMPS 2.0. Some tools may never be migrated over to GMPS 2.0. So there may be some tools in GMPS 1.0 that you really like. But for the most part, we're enabling interchanging of these different tools or of at least task IDs and all those kind of things. So it depends on what you're going to use. If you want to use for enrichment analysis, micromass, I would use GMPS 2.0.
Yeah, actually this workflow, the network enrichment, is only available in GNPS 2.0. So you can't run that correct. In GNPS 1. So another question, will GitHub repository be upgraded to GNPS 2? So there's another GitHub repository for GNPS 2. Another one was created. Okay, so let's continue with the parameters for the network enrichment. Hopefully this difference is clearer for you now. So the next thing that we will need to input is the GNPS networking task ID that I highlighted before. So if Shipei can paste on the chat, so you can copy that and paste in this field here in the network enrichment workflow. And then we are going to be selecting that this consists of a classical molecular networking job. So basically, it means that this data was not pre-processed before in other software such as MZMine or MS-Dial. So basically, if the files were pre-processed before, it would be consisted of a feature-based molecular networking job. But this is not the case. Please select classical. And then basically what we are going to do is to perform library searches with the spectra that are already deposited and part of the CMMC knowledge base that we are building. So all the parameters from now on are going to be parameters to perform these library searches. So the first two ones here, precursor ion tolerance and fragment ion tolerance, you can set that to 0.02. So these are parameters that are used when we are dealing with high resolution mass spectrometry data. In this case, it's quadrupole time of flight analyzer. And then you can set up the library minimum cosine to 0.6 and library minimum matched peaks to 6. Sorry, I missed how we get the network task ourselves. So in this case here, we already had a molecular networking job that was run in our previous workshop. So basically, if you have any molecular networking that you created with your files, you can use the task ID of that molecular networking job to run an enrichment analysis. In this case, it's already provided for you. How do you decide these parameters? Well, as I mentioned, in this case here for 0.02 on precursor and fragment ion tolerance, these are usually parameters that we we set for high resolution data, for high resolution mass spectrometry data. So basically, it is going to be comparing your spectra with a tolerance of 0.02 in the mass with the spectra that are deposited in the library. And in the case of minimum cosine, as I mentioned before, the closer that we get to 1.0 here, the closer that the more similar the spectrum will be. So this is basically a threshold of similarity that we will have between the spectra that are part of our data and the spectra that are deposited in the libraries. In this case here, the CMMC knowledge base. So it really depends on how strict you want to be here in this case. The less strict you are, the higher the chances of false hits. So usually we stay to between 0.6 or 0.7, but you can feel free to test other parameters as well. It really depends on your data sets. And here the library minimum matched peaks, it basically means, okay, I want that at least 6 fragments of my experimental spectrum match to the libraries, to the spectra that are deposited in the libraries. So at least 6 fragments should match. And the lower that you get here in this value, the higher the chances of having false positives as well. But this is also a parameter that you can change and see how the results look like. Okay, I think I'm going to continue for now.
Yep, I will let you know if there's a question.
Okay, great. So, and right here on top, results to report per query for library search, we are going to set it to 1. So basically what it means is that in many cases, what can happen is that when you are searching your experimental spectra with the libraries, you might have more than one match that will fulfill all the criteria that we specified before. If we just specify it to 1, and this is what we are going to do, we are going to leave it to 1, we are just going to see the one that has the highest cosine. But if you can put it, for instance, 2 or 3, so you can have a maximum number of hits per scan, per spectrum that you are searching, or of the maximum number would be the number that you select here. We're going to leave it to 1. And we are not running analog search. Well, let's keep this here to false, and we are not going to change here in the maximum analog search mass shift because we're not running analog search. So basically the analog search would be, okay, I want to search for all the spectra that will fulfill all the criteria above, but they don't necessarily will have the same precursor ion, basically. So now we can hit on submit workflow. And then you're going to be directed to a page like this in which you're going to have the information of your job. And it's going to be showing here on status running. I'm almost certain that for some of you, it will be showing status queued. It just means that right now in GNPS 2.0, there aren't many jobs that can be simultaneously run. So if many people hit submit workflow at the same time, some of you will be queued and will be run after the other ones finish. So right now, we know that this enrichment analysis will take about 20 to 40 minutes. It's not that fast yet. So we are already going to provide you a job that has already finished so that we can work together and explore how these molecular networks can be enriched based on this task that is already done. So if Shipei can please paste this job here on the chat, it would be great. And then you're going to see here that the status is done. Were you able to access this this job? Great.
I think the more important one is there anybody who didn't, that wants to
Yes.
follow, that did not get access to it? I think we're in good shape.
Great. So right now, what we are going to do is click here on download all results. And I'm going to wait a little bit because I know it can take a bit of time to download this file. It is going to be a compressed file. So please, for those who are following, if you can please let us know in the chat when you were able to finish the download, it would be great. So for those that say that, oh, it's queued, we are going to use the one that it was already run.
Yeah, because
before.
it takes 20 to 40 minutes to run. So take the link that Shipay provided in the chat, and we'll work with that.
Downloaded, yes, finished. Okay, I'm going to continue.
Laura has a question mark.
Yeah.
Laura Racina. It's still downloading, so give a few minutes for download. So,
yes.
We'll let you. We'll give you time. So not everybody has great internet speed, so this is. It shouldn't be that large of a file.
How large? It's about 80 megabytes.
Yes, it's a tar file, correct.
Tar file. Some people are getting an error accessing the... So you're having an error when you try to access the job link or when you try to download all results? Click download. The error. Okay, yeah, this is unexpected. So I'm going to send you here the file in the chat.
I just downloaded on my end and it's working, so not sure what the issue is, but indeed, if you can send them the file, that'd be great.
I don't know, I can send files here in the chat. Mm. 2.0 megabytes. Okay, let me just do something real quick.
Yeah, you can throw it in a Dropbox and then.
I'll do that.
Is it a TAR file?
TAR file.
Yeah, I got it from the link that Champagne sent.
I'm going to stop sharing real quick. I'm going to put in a Dropbox just in case.
maybe it thinks it's a denial of service attack.
Okay, so I uploaded that to Google Drive. So you can also try to download it from here. Oh, thank you, Felipe. You also sent
Yeah, and
it
also
here.
Claudia said it took a while for a few errors, but now it worked. I think it was just so many people.
Yeah, I think it's the first time that we are trying with that many people using GNPS 2.0. So yeah, let me go back to sharing my screen. Tar file is incompatible with Mac. No, it is. So okay, so I'm going to move on because right here I'm showing how in this case I'm using a Mac as well. So basically you can decompress this tar file and you're going to have a folder with all the files that are inside of it. So it shouldn't be a problem if you have Mac.
So I think we'll wait
basically,
one or two more minutes. Yeah, you can show them.
So basically, I just double click here, and it expands. I have here access to the folder, decompressed folder.
Yeah. Shipay or Felipe, could you link the decompressed folders for people so that they can follow along in the next steps? Because if it's an issue with their unzippers, essentially decompressing of the files that may not be up to date.
Could be.
Megan said, yes, Mac doesn't have a default for uncompressing tar files. So.
So Shipay or Felipe, can you please send this file at least like these two files that are small, which will be the CMMC enriched results, CSV, and the network.graphml. This is basically what we will need right now.
And Caroline Moore.
I'm working on it.
Okay, great. Thanks.
So
sorry about that, everyone. But
no, don't be. That's this is not this is a part of doing this kind of analysis. So it's a normal thing to happen. It's always about moving files, decompressing, using files. Can you read them? These are all a part of workflows. All right, Felipe posted it. I'll give you 30 seconds and then Helena can move on.
Okay. Okay, I'm going to continue then. You should have access to all the files that Felipe just shared here in the chat. So yeah, this tar file is a compressed file, and you should have access to all of these files here after you decompress it. But as I already mentioned, we are just going to be using just a few of them right now. So I'd like you to open Cytoscape that you installed previously. And please let me know when you have it opened. Opened, great. Great, so after opening, you're going to have a window like this. And we are going to see that we can basically just drag and drop the files, like networking files here in this area of the window. So what we are going to do is drag and drop the network.graphml file here into this page, into this window. So this file is going to be under gnps_network folder. And after you do that, you should have a window that looks like this. Everyone was able to open the networks? Anyone is lost? The folder is empty. So Solomon, perhaps you can check out the link that Felipe sent with the decompressed files. Okay. So Cytoscape is a very powerful software if we want to change the layout of this network. So as you get used to it, you will see that it's very powerful to build very nice figures for your reports, for your papers and such. So we are going to be following a series of clicks and changes of styles here so that we can really map the microbial information in a very visual manner for inspection. So what we can do is click here on this under the Style tab. And then we are going to have a change here in this part of the window in which we are going to have many different parameters that we can change for the visualization of these networks. Then what we will do for now is to click here on this fill color, which is going to be blue probably for everyone. And another window will show up with all the possibilities of colors that you can have for your nodes. And we are going to select here as white for now. And then click on OK. And another thing that we will do is click here on the border width that is set up to 0.0. And this other window will show up, and we are going to say that we want the thickness of the width of the border to be 10. And select OK again. And you will probably be seeing something like this that is kind of a bit faded. It's not really good to see. unless you can use really zoom in into the networks. So another thing that we will do now is click here on the top tab on view, and then select always show graphic details. And then you're going to see that this is going to be transformed into this, which is much better for visualization. Are there people that want to follow that are not following? And are there people that are following and that are seeing exactly what I'm seeing? Yes.
Excuse me.
Mm-hmm.
How to input the file, the task file?
So you can drag and drop that. Let me go back here on the slides.
Okay.
So it's the network XML file that I also saw that Mauricio sent on the chat right now. And you can drag and drop here into this portion of the window on Cytoscape.
Okay.
Yeah, and great. Thank you for unmuting and just asking.
Yes.
That's exactly what we hope people do. And the chat has been really great as well.
And just real quick, we should select here the style tab. And then in fill color, we should change it to white following these steps here. Are you able to follow, Tianyu?
Oh, yes.
Okay. And then after that, we are changing the border width here to 10.
Okay.
And then hit OK.
Okay.
And then you should select here on the View tab on the very top, always show graphic details, so that it's better for visualize the networks. Somehow I'm seeing the same as before, just black color.
I think it worked. I mistakenly chose the black color.
Okay, great. Great. Yeah, it happens. Okay, so we can move on right now. So now what we want to do is to import the information that was enriched by CMMC, by the library searches that we performed of the spectra that are present in the samples that were acquired from the samples, what they matched with CMMC. So we are going to click here on the top. There is this icon that says import table from file. And then you'll be able to select the file that you want to import. And in this case, you should select here on the folder CMMC results, this CMMC enriched results.csv. Oh yeah, Felipe or Shipe or Mauricio, if you can please send the CSV file in the chat, that would be great. Great, thanks. And after you select this file here, you're going to see another window that will show up that looks like this. And what you should do now is click here on this column called Query Scan. And this part here of the window will show up and you should click on this key icon. And then select OK.
Is that like a filter or something else?
So it's basically, we're importing more information. So we are going to have more columns available to perform any kind of visualization that we want in Cytoscape.
And the key is to let Cytoscape know how to map, like how to migrate each row to the exact row in the previous network, because we're just mapping information on top of the network. So this is just to be able to link this new table with the previous table.
Thank you both.
Okay, so right now, we are going to have more, we will be able to map some of the information that we just imported to, for instance, fill the network, fill the nodes of the networks. So now what we will do is, in this part here, fill color again, that is all white for now. What we want to do is to color just the specific nodes. So we can click here in this arrow, and if you click here, you're going to see that that this column and this mapping type row are going to show up. And what you can do is here on the column, select input source. So it's one of the columns that were imported from the CMMC results table. And here on the mapping type, we can select discrete mapping. And then you're going to see that two other rows will show up in which we have the information if it's microbial, if a compound is microbial, or if it is microbial and a diet-related compound.
Elena, can I ask you something?
Oh yeah, sure.
This mapping type by microbial or microbial and diet is something that the database that you import has already, or
yes.
Oh, okay.
Yeah, so later on, Peter will show very briefly what are the types, what are the fields of information, of microbial information that we are going to be adding. And this source field is one of the fields that people have already deposited and that we can map here.
I see. Thank you.
Can I ask a quick question about microbial and diet?
Yeah, sure.
Is microbial and diet means we don't know if it's microbial and it could be diet, or it means it's in both microbes and diet?
In both microbes.
This is a great question. So in this case, it's found to be or the way we classify it in this case is that it's found to be associated with or found to be produced by microbes or can come from diet. And I'm thinking about, let's say, essential amino acids that a human cannot make, but has to come from diet, or vitamins such as thiamine. Microbes can make it, or it has to come from diet. Those are the kind of molecules that would be classified as such. We also have other classifications where we have microbial metabolism of food or microbial metabolism of host-derived metabolites. So those are different categories.
Yeah, so basically right now we are going to be mapping many different information. This is the first one that I'm showing. So all of this was already curated and deposited in CMMC.
Sorry, can I ask a related question of that? So, Peter, you said that you check metabolites that are being produced here in this case. Can you also have do you have also the information of metabolites that are being consumed by the bacteria?
So we do capture that information in the knowledge base. So in principle, it could be it should be a part of the file that's incorporated into the Cytoscape file. So we do try to capture that, and the reason for this, why this is nice, is because if some of the microbial metabolism or metabolites are not a part of standard metabolic pathways, you need to start building new metabolic pathways, and that would permit that. So the precursor substrates are documented if people upload that, and then the products of those reactions are documented.
Okay, thanks.
Yep.
I have a related question too. Is there any parameter for diet only, just to subtract the effect?
No, we have not built diet only. So it's something that we should think about adding, but the focus of CMMC is microbial metabolites. That's why we don't have an option for diet only. But we do have something called food mass. And so here you can search against, at this point, it's 3,600 foods. And you essentially get information of what foods that specific molecules is found in, but it doesn't tell you whether it's also found to be produced by microbes. So in principle, we could create a food-centered curation effort where we indeed add food-derived only metabolites that are not made by microbes. But that's not something that we've added at this point yet. But what I would do is I would search all the MSMS spectra against foodomics or the food data sets that we have with food mass. Overlay that onto the networks that you're creating here so that you can see which ones are likely associated with foods.
Thank you.
Oh yeah, and,
Yep.
and Shippei also sent the paper for
Yeah.
FoodMast.
Yeah, so food mass, the paper is there. It's an old version of of mass searches. It will take 20 minutes or 40 minutes per MSMS spectra. We have a new one where it takes seconds per spectrum to search, so you can search entire data sets now.
Okay, so I'm moving forward.
That's a great question.
Yeah, all of them are very good questions. So right now, as you can see, we have two rows here about microbial and microbial diet, and we can select both of them here and right-click here on this part of the window, and you're going to be able to select here mapping value generators. And there are several palettes that you can pick up, but I would recommend color blue or dark colors. And then you're going to see that just some of the nodes were colored. So these are basically nodes that matched with this particular classification of microbial or microbial and diets here in that specific column that we imported from the CMMC results. And we can see here that there is a beautiful molecular network that was that many of the nodes were colored here, that it really pops up. And what we can do right now is see, okay, I want to see what are these compounds, because these numbers, they are not informative right now. So we can click here in the label row. We can click here in this arrow again, very similar to what we did with the fill color. And here in column, we should select input name. And in mapping type, we can put pass-through mapping. So basically everything that will have input name will appear as labels. And we are going to see that all these numbers here, they now have labels, they have names of the compounds that were deposited in CMMC. And what we can see here is that this part of the network is relative to cholic acid derivatives, and this other part here to the oxycholic acid derivatives. So basically, these are bile acids conjugates that conjugated with different amino acids. As I briefly mentioned before, this data set consists of two human fecal materials and also microbial cultures that were cultivated with unconjugated bile acids, so just with cholic acid and the oxycholic acid. and also the amino acids. So what we see is that these conjugations here were shown, and they were already deposited in CMMC as well.
A quick question here. So
Mhm.
the two kind of clusters here that are connected with one node in the middle. I mean, I know it's not shown here, but would that node, for example, would be the parent, I guess, bile acid, like the deoxy or cholic acid, or not necessarily?
Not necessarily. Not necessarily. You would need to manually inspect each of these nodes. So this type of mapping is very powerful, because right now we can see that we can see all these microbial modifications, and what we can do as well. We're not going to explore that here right now, but what we can do is really try to propagate this annotation based on the difference of the precursor ions between two nodes. So then you would need to really manually inspect, okay, what could be here?
Yeah, so,
if it has an annotation.
Yeah, I think I can give a slightly clearer explanation. So originally, some of these new microbial metabolites, such as these bile acids, where you see the R is modified differently. So typically, we knew about glycocholic acids. Well, it has a glycine attached. But then if you look at maybe some of the deltas, so one of the edges, and let's say it's 90 Dalton, 90 point something something, that based on my experience in proteomics, I knew there was a glycine to phenylalanine substitution. And so you can start to inspect the edges in relationship to something that's annotated and try to predict what kind of modification there is. So if you see 14 Dalton, that's probably a CH2. Usually it's a methyl group, but if it's a fatty acid chain, it could be an elongation of the fatty acid chain, for example. If it's 15.995, it's an addition of an oxygen. And so you can begin to start inspecting and propagate some of these annotations. And so in this case, you see that these bile acid conjugates, you have cholic acid plus alanine, threonine, a lot of the amino acids that are there. More recently, we've discovered that there are tyramines, diaminobutyric acid, or GABA, some of the polyamines that microbes are conjugating to these bile acids. So bile acids is just one example, but there are lots of other molecules that fall into these kind of categories, and it really helps you to propagate the information that you have.
Hi, can I ask maybe another question?
Yeah.
If I want to check from which bacteria do these metabolites come from, all this information, it's in the enrichment analysis file, right?
Yeah, we'll get there.
Okay, sorry.
Yes.
No, it's perfect. Great
Yes.
question.
Okay, so just going to highlight that there is another node here that popped up as a compound that is related to microbial and diet. And basically this is a dipeptide. But all the other ones here that appeared before were just linked to be microbial compounds. So another type of mapping of information that we can do right now is go again to field colors. And we are going to change here the column that we will map. So we are going to select now input molecule origin. And again, we will select discrete mapping because we just want to color some specific nodes, not all of them. And we are going to select all of them again and right click, go to mapping value generators and select again color brewer dark colors. And now we are going to have a different type of information here. In this case, we're going to have the information if the compound was de novo biosynthesized by microbes, if it is a microbial metabolism of food molecules, or if it was a microbial metabolism of host-derived molecules. And now we are going to have a network that looks like this. So all the bile acids that we saw before, they are classified as microbial metabolism of host-derived molecules, basically because these unconjugated bile acids are produced by the host in the liver and then further conjugated with microbes. And again, here that compound that popped up as dipeptide that popped up as microbial and diet here, it was mapped as microbial metabolism of food molecules. And we also see one here that it was classified as being produced de novo by microbes. And in this case here, it's C-trioviridine. And yeah, so basically it's another type of information that you can map into the networks. And of course, that we end up asking the question, okay, so which are the microbes that can be producing these compounds? That's Claudia already asked. So let's do it right now. Let's get a list of the microbes that can be producing these compounds. Let's go again to fill color, and we can select here in column input microbe name, and again discrete mapping. And we're going to have here lists of different microbes, and we are going to select all of them here and right click again. But right now, I mean, since there are many different combinations here in these lists, let's just edit. Go to edit and edit selected discrete mapping values. And we are going to see again this window. And let's say that we want to color all of the compounds that have a microbial information in the sense of which microbes that can be producing them. We're going to color all of them in as green. And then select OK. Now, again, all the compounds that matched with the CMMC libraries and that had the information of which microbes could be producing these compounds are mapped here in the network. So let's go back to our bile acids network. And what we can do now is, for instance, click in one of these nodes that are colored as green. For instance, here I'm clicking on the oxycholic acid conjugated with ornithine. And in the table that we have below Cytoscape, we can go to the right and find the column input microbe name. So right here, we are going to have a list of all the microbes that have already been associated to be producing or these compounds, for making this transformation here.
Could I ask a question about the previous part with the microbial, with the origin, I think, with the source?
Yeah, sure.
Like, I noticed one of the notes, for example, was the indole lactate or pyruvate, I think.
Mm-hmm.
And I guess my question, because it was labeled as it's coming from a food, I guess, or a microbial metabolite from food. I was wondering, I guess, how do you make the judgment whether it's, I mean, since tryptophan also can be made by some bacteria, how is it linked there, or how are the decisions made to label it from food more than?
So this is where we can have a discussion later on this, but this is a discussion where basically it's somebody who curates the knowledge base will provide that annotation. And so it's really the user that decides this. Now, there is a way to edit what somebody else has provided so that you can enhance that information. But the knowledge base is as good as what the public is or what the community is able to provide. So it's a great question. I mean, in this case, I think the majority of tryptophan comes from diet. I think relatively little comes from microbes, but on the other hand, you can't exclude it. There's going to be lots of these kind of gray areas and we started to see some of those as we built a knowledge base, that it's not always as easy to define as we'd like.
Yeah, no, that makes sense, and I guess it will be, I guess, impossible to make a new category for each of those murky. Yeah, thank you.
Correct.
Yeah, but it's a great question. And as Peter will show later on, we also have a field for references. So people who are curating this information, we will also have links to the references that support all of the information that was input.
Correct.
So yeah, now I have shown you how you can get the list of the microbes that can be producing these compounds. But well, before moving forward, I would like to ask you a question actually. Considering the data sets that we have, fecal material and also cultured microbes, would you, I mean, do you think that this coverage that we are having here is big? I mean, would you be expecting to have perhaps more nodes that are populated? Yes. So basically this shows that we really need a community effort to deposit information in the CMMC knowledge base so that this type of analysis can become more and more powerful over time. So right now, I think that most of the compounds that we deposited here in the knowledge base came from our lab, and we are doing a very good job in with the bile acids in particular. It's being one of the main focus in our lab. But of course, there are many other classes of compounds that need to be uploaded to the portal so that this type of analysis can be even more and more informative.
Yeah, we'll get to that when I highlight the community call.
So we can very quickly as well explore the list of microbes that can be producing these metabolites. If we go back to the network enrichment job where you downloaded the results, there is another link here called CMMC search results that you can click. And you're going to have a table like this. So here we are going to see all the compounds that matched with CMMC knowledge base and all the microbe names, the microbes and CBIs and the source of them. It's not complete. You're going to see as well with Peter that there are much more information that we're capturing. We are also capturing activity data. But here it's just a very brief overview of all the compounds that matched with the knowledge base. And well, since it's Halloween, I'd like to give you not a trick, but a treat. I'd like to give an activity for you to work on. So now you have learned on how you can map all this microbial information in Cytoscape if you have already a network enrichment workflow. So now you can try to do that with a new data set. We are going to give you about 15 minutes, 15 to 20 minutes to do that, and you can answer some questions. So this is another data set which consists of human fecal material from a diet study. So basically a group of people are vegan and the other group is omnivore. And this fecal material was processed and analyzed by LCMSMS. And we already had a molecular networking job for this specific data set, and we already enriched it. So the enrichment result is here in this link. So if someone can please post that on the chat. And well, you can answer then the following questions. Which of these compounds are a result of microbial metabolism of food molecules? Which of them can be produced by de novo biosynthesis of microbes? And which of the microbes have already been described to be producing these compounds?
Yeah, and so we'll at 10:35 San Diego time, so basically 35 after the hour, then we'll come back. But you can ask us questions while you explore these 3 questions yourself. I always find that exploring on your own really enhances the learning and what can be done and what kind of questions you might have. And then at 10:35, I will highlight the call to the community. So in 14 minutes. So feel free to ask questions, anything, even outside of this workshop. We're happy to answer a question about GMPS, molecular networking, or anything else.
And just another note that I would like to do is that in the CMMC website, we have a tab here for the documentation. So we have very detailed documentation about, for instance, the network enrichment workflow. So all the steps, the step-by-steps that are necessary to do this type of analysis, and also how you can deposit data. Peter will show you after 14 minutes. So yeah, if you have any questions as well later on, you can check out this documentation, and it is also a work in progress. So if you think that there's something that is not clear in the documentation, also feel free to let us know so that we can improve it.
Yeah, we often, people contribute to help with the documentation. So if there's something not clear, you essentially help us correct it. And then you're also, we welcome you to be a co-author of the paper as once we developed this workflow system. Can you go back to the questions so that they can focus on the questions? Someone has trouble downloading the files. Okay, Felipe will share the files. Thank you, Felipe. I don't know what the size of the latest file is. Sorry. Felipe can tell you.
Felipe, I can't seem to find the network graph ML file, which I think I'm trying to look for. Are you there, Felipe?
Yeah, it should be in the GMPS underscore network folder. Do you not see it? I can see it here in Google Drive. I can actually, let me just share that specific file directly on the chat.
Yeah, share the file because there are some issues similar as the previous one. It is not consistently downloaded for everyone, I
Yeah, it's
assume.
basically too many hits to the server at once for FTP downloads, which is great. So we'll take that in account in future workshops. This is the first sort of workshop where we downloaded things from GMPS 2.0. So we're learning as we go along.
Yeah, I just put the network.graphicml file in the chat, and then let me just also share the CMMC enrichment table. I think that's pretty much what you need.
May I ask, hello?
Yeah, of course.
Regarding the online visualizer, it's nice that we can have the spectra at the same time. There is some advantages of Cytoscape apart from the nice colors that we would prefer, or we can work online because I see that we can overlay also the data that we were working on Cytoscape, and we
Yes,
have
this.
the spectra as well, which is great.
Yeah, this is very new. I think it was deployed yesterday or 2 days ago. So we had prepared before with Cytoscape, and like Cytoscape is more customizable as well. So
Yeah.
but you can also explore that in the browser. But we are still also testing this.
Yeah, GMPS 2.0 is undergoing many changes rapidly. So
yeah.
But in the end, I think I'm in agreement with Elena. In the end, things such as Cytoscape, there's more customizability, there's more integration of additional data if that's needed. But the GMPS 2.0 will be a really nice way to quickly navigate things.
Indeed, the only very good thing is that you can visualize the spectra that I see now that you cannot do on Cytoscape because it's
maybe if you can show people, it would actually be kind of cool to see for people what you're looking at. You're welcome to share your screen.
Yeah, actually I cannot because I'm working on different desktops. But
never mind.
Yeah,
that's a pity. But either way, it's nice. All right, Helena, you can show your screen. But yeah, there's so many ways that we can work with this data. But this is just a starting point. As I said, we're just barely crawling, and this is. The way I think about this is when we started even doing molecular networking in 2012, that's sort of the same feeling I have with building this knowledge base and using this knowledge base. It is very early, and I know it's going to be very useful and powerful in the long run, but it's already becoming useful. So Elena, if you put up the questions again, that would be great. And it's also very similar to when we developed GMPS 1.0. By the time we submitted the paper, there were already 80 papers that were using the GMPS platform before we even submitted the GMPS paper. And that may happen here as well. So Vanessa, you're welcome to share your screen if you wish.
Okay, yeah, I'll go ahead and try.
I was just gonna answer that question on the chat actually. Cytoscape
That's
is gonna
okay. Let
show
her
you.
share the screen. Let her share
Okay.
the screen. Okay.
It does say host disabled participant screen sharing on my end.
Oh yeah. Give
I
her.
I will. Yeah.
Give her the power.
Okay, you can go ahead.
Okay, perfect. Let me find the correct one. Okay. So as you see here, like for the mapping type for fill color, I put the input molecule origin and then discrete mapping, but I only got the de novo biosynthesis by microbes and then microbial metabolism of food molecules. When last time, I believe we had also microbial metabolism of host derived, whatever it is finished.
Yep.
So for this, would it be correct if we only got 2 this time around?
Yes, this depends on your
2
data
of the
set.
categories, yes, it depends on your data set for sure. You also see how sparse the annotations are, but on the other hand, the only option you would have had to get that information would have been Googling every single one of these metabolites and see if you could trace the origin. If you've ever had the pleasure of doing that, you'll know how long that took. And
yeah,
this is one of the
time.
Yeah, go ahead. What were you saying, Vanessa?
I said that just did not sound like a good time trying to Google everything.
Exactly. And so this is one of the reasons why we have this call to the community. One, we now have a tool in place that we can actually leverage the knowledge base. So you can learn about microbial metabolites quickly if it's in the knowledge base and it's in your data.
Okay, that
The
makes sense.
next part of the workshop will be, I'm hoping that many of you will want to contribute to this knowledge base, basically.
Okay, that makes sense. Thank you. I appreciate it.
Yep. And thanks for your willingness to share.
Of course.
All righty. Does anybody have any questions about this? If somebody can answer Jessica's question, because I'm not entirely sure.
Yes, you can have several networks open at the same time. If you go to, actually, I'm gonna share my screen.
You need.
No, I do have permissions.
Okay, great.
So if you go to this part here, we were here in style. This is the previous network, not for this second job. But then if you go here to network, you can have several networks here. See, I only have one, so one of one network selected. And that basically implies that you can have several of them here.
I see. Thank you. Because when I add the second one, I don't know why, but on style, I cannot select the input by microbiome name or molecule origin or something
Well,
like
remember to be able to do that, you input the network, but then you have to import the table. I also shared the table in the chat. So maybe you haven't imported the extra table that adds those attributes that you can later use here in the style.
Oh, yeah, that's true. Thank you.
You're welcome.
No, that's perfect.
I'm also facing this issue. I did input the table, but I'm not seeing any label on the notes.
Maybe if you share your screen, if the same issue exists,
Yeah.
we can give you feedback.
Can I get the permission?
Yeah, you can share now.
Thank you.
We can see your screen now.
So I did put the table from here.
Mm-hmm.
This is the result table I got. And I did put the key scan and okay. But when I'm going to label, I'm putting input name or anything. This is the problem I'm facing.
So you in this case, you can select pass-through mapping because you want to put the label in everything that has a label.
Okay.
So if you zoom in, for instance, in that purple one,
yeah,
you should see the label there. Yeah.
But there's no label. Okay. Yeah.
Yeah, there's just very few labels.
Yeah.
Yeah, there is. Yeah. Okay,
Good.
thank you.
Great. I think I will now go to the last part of the workshop, and that's more almost the call to the community. And this will be the first time we're creating the call for this particular thing, for this particular topic. We've had other calls. So for example, Microbial was a call to the community, and I think there's about 110 authors on that paper that's contributed to Micromass that allowed us to curate 60,000 LCMS runs taxonomically. What we're hoping to do is something similar here. Something similar here, but now to curate microbial metabolites within the ecosystem. So you saw this slides before when we started the workshop that you were going to learn about the network enrichment workflow, which you now learned about. But the network enrichment workflow actually draws the information from this microbial metabolite knowledge upload portal. And when you looked at your networks, you saw how relatively few number of microbial metabolites are currently annotated. And that's just because the microbial metabolite knowledge upload portal only has about 600 metabolites in it, although it has about 5,000 MSMS spectra associated with it. And so the idea is that what we're gonna begin focusing on over the next few months is call to the community to populate microbial metabolites. These could be microbial metabolites they've discovered themselves, or from papers that have already been published in the public domain, and they want to add these different metabolites to them. We would like people, if they're interested in contributing and really growing this knowledge base, so to speak, we would love people to contribute, because that is what is allowing us to figure out what are microbial metabolites, if it's food that is metabolized, etc. So the way I sort of view it is that we have this black hole that we're trying to basically suck up all data and build a knowledge base with it. So the knowledge base itself, you'll need your GMPS 2.0 login to use it. But it contains data selection, so it has a data selection option. And this is done via the Universal Spectrum Identifier, and I provided a preprint link above it. But basically any data set, any MSMS spectra that's in the public domain, it doesn't matter whether it's in the repository metabolites, it doesn't matter whether it's in Metabolomic Workbench or GMPS Massive, you can create a universal spectrum identifier for it. This is also the one that we feel is required, because we actually want to connect the microbial metabolites to raw information or raw data so that we can reuse that data. It's very similar in the sequencing world, where if you want to identify an organism, you link it to the sequencing. So that you can reuse that sequence information to find other that same metabolite or same sequence in other data sets. And we have detailed instructions on how to create universal spectrum identifiers, all these kind of things, how to deposit data and where you can deposit it. We have all that information. But a part of the knowledge base, and you have to think about the knowledge base we won't have every piece of information that we want about a microbial metabolite. And so microbial metabolites, you can provide as much information as you can that is possible. But if the molecule name is known, put it here. The SMILES or in-sheet partial structure, you can add that to the knowledge base. And we literally have an upload portal for this. So then we have this drag down option of molecule origin associated with the metabolite identification. And this is really about the microbial metabolism of different molecules. And so you have microbial metabolism of drugs. There's a group of people that are trying to curate some of these now, so that if you encounter a drug in your data set and it's derived and it's being metabolized by microbes, that we start to be able to see those. Microbial metabolism of food, so the tryptophan to some of the other tryptophan-derived metabolites would be a nice example of this. Microbial metabolism of other human-made molecules, and so we're thinking about things such as PFAS, so the polyfluorinated fatty acids. Maybe some of those are microbial metabolites. Maybe there's some plasticizers that are metabolized, etc. But essentially, non-drug-like metabolites that are made by humans. Microbial metabolism of host-derived metabolites, that makes a lot of sense. Microbial metabolism of microbial-derived molecules. And then we also have host metabolism of microbial metabolites. And so something such as 4-phenylethyl sulfate, where the sulfate addition to 4-phenyl ethyl is a host metabolism of microbial metabolites. And so we added the categories of this. De novo biosynthesis by microbes, we're more thinking about the natural products such as penicillin, rapamycin, vancomycin that are made by microbes in that fashion. And then whatever is unknown or undefined. If there are additional categories that people can think of, you're welcome to provide suggestions as you go through the process. We have a taxonomy and phylogeny selection. There was a question about this earlier. I like the idea of calling it synthetic. That's a fair point. Taxonomy and phylogeny. So taxonomy is not necessarily phylogeny, but most people in the world actually use taxonomy. However, microbiome field tend to use phylogeny. So if you have taxonomic information and it matches to NCBI, add the NCBI number. If you have the microbe name, put as much information as you have. But for the people who do sequencing, if you have sequence data associated with these organisms, I would put the sequence information in there. And then if it comes from a microbial community, put the link to the sequencing information to the microbial community and the community description. So this is to accommodate both taxonomy and phylogeny. And of course, not every metabolite is going to have all this information. And so this is why we have these different options. Then in terms of biosynthesis, there's something called MIBIG, where people catalog microbial metabolites and the biosynthetic information. If it's in MIBIG, great, that's awesome, add it. But we recently got a paper accepted with the Patterson Lab, as well as another group back-to-back. They will come out back-to-back. We found that bile salt hydrolase is actually doing the cross-coupling of these amines to bile acids. Well, that's not an annotation in MIBIG, but then you can put the input biosynthesis genes here and give a biosynthesis description so that we can start to think about building new metabolic pathways associated with microbiome-derived molecules. And then we also have the microbial metabolite source. This is, you can click that it comes from microbes or microbes and hosts, diet, microbial host and diet or unknown. And then there's an entry point where you can put in the precursor structure and the end structure as well, so that that can be documented as needed. And then if there's activity information that you know about, so maybe it does T-cell differentiation in IL-17, or maybe it's antimicrobial activity, maybe other activities that can be defined, we'd like to capture that information as well. And we, for now, give it pretty broad categories in terms of activities, in terms of how you fill it out, because we do not actually know what people are going to add in terms of activity. But the nice thing is once you have a microbial metabolite that matches, you can check, is there any activity that is known about those microbial metabolites, which is really useful. Caroline said it's heavy, I think there's a couple of questions. This is heavy on microbe hosts, or is it this where other microbes, cyanobacteria, will also end up? It can be any microbial source. So if you're doing drug discovery from natural sources, it's fine to add those activities here as well, because it can actually help. Yeah, like Michael M, synthetic may be better. I think we had a reason for it. I can't remember the reason why we did not use that, but I do like the idea of synthetic. And then grant information, comments, and of course we want references. If you have references where this has been documented so that people can actually go and check those references, that's really useful. Preprints are fine. Other references where some documentation in terms of this information that you provide is very useful. So then to access the knowledge base, there's a link here to access the knowledge base. You can actually download it, but basically there's two forms of download that you can have. One of them is focused on the MSMS spectra. So if you want to download all the MSMS spectra, link to the different compounds, you can click on this download USI database, and you can get all the information that's attached to these particular compounds. Here you can just search if you wish based on a compound name, microbe name, or user. There's also the structural entries. If you want to search based on SMILES, you can do that as well. So basically the idea is that anybody can download the database and use them in third-party tools if they wish, just to make it easy for the community. And as you can see, we have about 478 pages of MSMS spectra now, but you can clearly see some of these are duplicates. So it's about close to 5,000 MSMS spectra that have been curated, but it's only about it's about 600 compounds at the moment. Now, what we're doing is we're beginning to write the write the paper. This may take a half a year or longer, which is typical for these kind of papers. But if you're interested in contributing to this paper, I would love you to put your email in the chat and your name. Then some of us will contact you in the next couple of weeks. But essentially, we want people to help curate the knowledge base. We want people to test and document use cases. So one of the things that we often like to do is basically showcase how a tool or a knowledge base that we develop, how it's useful to the community. And the way we do this is by having the community actually test and document some of the use cases. We've done this many times. We do this for many other tools. And what we get back out of it is that you give us feedback in terms of what worked, what did not work, how to improve. And an example would be describing, maybe we should describe this as made synthetically versus human made. And so those are the sort of different discussions that we have. And my hope is that we will get a few thousand compounds to curate in the knowledge base over the next 6 months or so, so that it becomes even more useful than what you've already seen so far. Anyway, anybody has questions, thoughts, comments? If you're interested, paste your email in the chat. We will record your email and then approach you. Basically, the options of how to help contribute to the knowledge base, if you have papers or you have microbial metabolites that you've documented yourself and published on, those can be added. If there's literature papers on different molecules that you think should be added, we should definitely add those. If you have new unpublished compounds that you might be interested in adding, we would love to see those in here as well. That's it. You can unmute, ask questions, or in the chat. So there's a couple of questions. The host microbiome metabolomics facility team at University of Chicago is interested in contributing. Okay, great. Kayata Kaider, do you have a database of disease versus healthy? No, we do not. However, let me show you, because I have that at the end. So right now, what we're doing is the microbial metabolite knowledge base on the left, and we're capturing structural information, source of molecules, and some phenotype of activity information. And we link this to the universal spectrum identifier. And then we have this knowledge base that can be downloaded. Now, there's another tool that we're building at the moment, and these are under construction, but similar to MicroBMAST, where we can search with an MSMS spectrum and find the taxonomic distribution, not phylogenetic distribution, but taxonomic distribution of the microbes that are part of that database. So there's 60,000 LCMS files that are part of this. But the other thing that we're doing is we're curating data sets from organisms, environmental or observations, but also disease states. So in other words, what we're trying to do is build something similar to MicroBMAST that you can see if a specific microbial metabolite is associated with these different health conditions or conditions. And these could be, it doesn't only have to be humans, it can be animals. It could also be ocean environmental conditions where you have, let's say, a cold upwelling in the ocean. We're trying to capture all this information, but of course a big focus will be on human health conditions and animal health conditions, interventions, all those kind of things, so that you can begin to map these observations. Kayati, did that answer your question?
Yeah, it does. Is there any way also to determine the concentration, whether they are high or low? Fold change or something?
So we have not, so the current one will be either observed or not observed in these different phenotypic observations. I think one of the things we want to develop in the future, but that will be at least a year away, is indeed add quantities like you're describing, where we will create box plots out of these things. What these mass sort of tools allow you to do is identify the data sets where you putatively have changes, because in principle, the frequency of detection is related to the intensity of the molecules. It's called spectral counting, but it only will find really large changes. And so what you can do is you can use essentially to find data sets where these metabolites are found. And then you can go back and reanalyze those data sets, do feature extraction and all that, all those kind of manipulations. And then you can draw your box plots or do your statistics on it to see their relative intensities. So it's not as easy, and it will not be that straightforward. But on the other hand, without these search tools, you wouldn't even know that those data sets existed.
Yeah, we have some data from like celiac disease, type 1 diabetes, and we are actually comparing this changes after the LCMS.
Yep.
So that's why I was thinking if there is a whole database, that would be, yeah, great.
That's perfect. Yep. Yeah, so there will be a couple of different diabetes data sets. There's soon going to be a diabetes data set of infants. So essentially newborns, and some of these new microbial bile acids tend to be really good diagnostic markers for prediction whether an infant is going to develop diabetes. Oliver Barr is interested in contributing siderophores. That would be great. So there's a couple of studies ongoing in that as well, but certainly it will be wonderful to add. You can search it with and without the metal additions. I don't think many of the siderophores that we know exist have been annotated already in the knowledge base, so this would be a great contribution for this. Thanks, Claudia, for great tool and resource. Let's see. Benedict Geyer, related to infectious diseases, that will be really fantastic. Alrighty, that's it. If you have any further questions, let me know. And that's the end of the workshop.